Good evening, everyone. Welcome to the last of a three-part webinar series on COVID science, hosted by the Office of Lifelong Learning and the Physician Learning Program at the University of Alberta. My name is Denise Campbell-Scher, and I'm the Associate Dean of the office. Our office is committed to providing reliable advice and resources for healthcare providers, scientists, and everyone committed to addressing the challenges related to the COVID-19 crisis. I'd like to review a little bit of housekeeping for this evening's webinar. So on the bottom of your Zoom screen, there are a couple of features. The Q&A feature is the feature to use if you have any questions for our panelist. And the chat feature is if you have any technical questions for the staff of the office, or if you have any questions about L3 services. Dr. Houghton will answer several questions following his oral presentation. He anticipates being finished around 7:40 to 7:50. Some questions will be responded to through text in the Q&A window, and he will answer questions until 8:15. You can at any point use the Q&A button at the bottom of your screen to submit questions. We anticipate many questions, so please review what's already been asked and upvote those questions that are closest to your questions and that you would like to have answered. We cannot guarantee that all of the questions will be able to be addressed. A recording of the webinar will be available early next week. It will be sent via email to all those that are registered. The recording will also be available through the L3 website and our social media channels for those that did not register. So we are very pleased tonight to be able to introduce Dr. Michael Houghton, who will be sharing with us his perspective on prospects for COVID-19 vaccine. Mike is the director of the Li Ka-shing Applied Virology Institute and is a professor in the Department of Medical Microbiology and Immunology at the University of Alberta. He is an international leader in viral hepatitis. In 1989, his USA laboratory and collaborators identified the hepatitis C virus and then developed blood tests that have since prevented millions of infections from tainted blood donations around the world. He has received numerous international prizes for this work, including the 2000 Clinical Lasker Award, the Robert Koch Medal in 1993, and the Canadian CASL Gold Medal in 2012. He leads an experienced team that's been developing a vaccine against the hepatitis C virus and now intends to use the same technology to expedite a COVID-19 vaccine. Thank you very much, Mike.
Thank you, Denise. Hello, everyone. It's very nice to be talking to you this evening. Trying to get the next slide. I can't forward the slide anymore. There we are, thank you. Well, I'd like to jump straight into vaccines that are looking promising. And the first one is from a company called Sinovac in China. And they published their work in Science just a few days ago. And the vaccine is actually an inactivated viral vaccine. So they make this in viro cells. They infect viro cells, which are African green monkey cells. They then transfer the cells to a cell factory to get high density cultures. They then take the supernatant of that cell factory and they inactivate the virus with a chemical called beta-propiolactone. Following careful analyses that all the virus is dead, they then concentrate the virus, treat it with benzonase to get rid of any nucleic acids outside of the virion, and then perform purification on size exclusion chromatography. They filter the product through a 0.2 micron filter to get rid of any bacteria that might have got into the product. And then that is the inactivated viral vaccine, to which they add aluminum hydroxide as the adjuvant. And that gives them the vaccine product. Just trying to get my cursor to show on the screen. Anyway, in panel D, you'll see on the left-hand side some molecular weight markers. This is a gel that has been electrophoresed. And on the right, you can actually see the product. And you can see right at the top of the gel what is the spike protein of the virus. This is the protein that sticks out from the surface of the virion. On panel E, you can actually see the purified product, the concentrated virus particles. And on the outside, you can see the spike protein. And it appears often as a corona, as if you're looking at the sun, and hence the name coronavirus. So this, as you can see, quite highly purified virus contains the spike protein, the nuclear capsid protein, and then the matrix protein. So this is a fairly pure preparation. And they've immunized rhesus macaque monkeys. They immunized the monkeys 3 times on week 0, week 1, and week 2 with either 3 micrograms or 6 micrograms of the vaccine. And then after immunization, they challenged these monkeys by actually introducing the virus into the lungs of the animals on week 3. So they challenged 1 week after the final immunization. And what we can see are the results on this slide. I think perhaps the most important one is right at the bottom, panel F. What you see on the extreme left panels are the pneumonia, the severe interstitial pneumonia in animals that did not receive the vaccine. Either they got a placebo or they got a sham, which means the adjuvant but without the vaccine component. On the other hand, when you look at the lungs analyzed from the vaccinees, shown on the right, you can see that the histopathology was either normal or at worst, there was a mild interstitial pneumonia in these animals. So a drastic effect on the disease. If we look at the viral load in these animals after challenge, you can see in panel C where throat swabs were taken and the viral RNA was quantitated. In the placebo and sham, you can see that the viral titer was approximately almost 10 to the 6 copies per mil in the throat swab. In the 3 microgram dose vaccine, you can see that it gradually diminished over the week following challenge. And in the 6 microgram dose, you can see an even more pronounced reduction in viremia in the throat swab in the following 7 days. The anal swab, you can see the placebos once again had high levels of virus, 10 to the 5 per mil, but with both doses of the virus, there was no virus in the anal area. When we look at the lung in panel E, you can see that the placebos, the green and the gray, obviously had a lot of viral RNA in the lungs. They probed the left lower, the right lower, middle, and so forth. And you can see that there was 10 to the 4, 10 to the 5 copies of virus per mil in the lungs after challenge. In contrast, both vaccine groups failed to show viral RNA in most of the specimens taken from the lung. On the far right, you see that in the right lower lung and also in the right accessory lung area, there was a small amount of viral RNA in the low dose vaccine group. This is a log scale on the left, so this approximates to around 10 RNA copies per mil, in contrast to 10 to the 4, 10,000 or 100,000 copies in the controls. So this is very nice evidence that this vaccine protected these animals. And there has been some concern with past coronavirus vaccines that some actual toxicity can arise and not the prevention that is required, obviously. And so the histopathology examinations of the lung demonstrates that not only was it effective, but they did not see any adverse events in the form of eosinophilia occurring in the lung. So this was very promising data published very recently. Next slide, please. Thank you. Yes, so with a vaccine to COVID, there are two important parameters. One is obviously to prevent disease, but as I said, it's also to make sure that we don't see any enhanced disease, either from a virological point of view or from an adjuvant point of view. And they looked at this quite closely. Panel A, they're looking at T-cell subsets in the monkeys to see if anything happened. Cytokine storms are associated with disease in COVID, and they were looking at CD3, T cells, CD4s, and CD8s, and they could see no difference in these animals, in the vaccinated animals versus the controls. When they looked for cytokines in the vaccinated animals versus the controls, there was no difference in circulating levels of TNF-alpha, gamma, or IL-2, which are all inflammatory cytokines. And then when they looked into the lung tissue of these animals, they saw no evidence of any kind of Th2 augmented toxicity. They saw no eosinophilic hyperreactions as a result of the vaccination. So this was very good data. Next slide, please. Now, importantly, they also asked the question, does their antisera to this vaccine neutralize virus taken from different parts of the world? And the best panel to show this are panels D and E, where they're taking antisera and they're looking for the ability of the antisera to neutralize the infectivity of different isolates. There were 3 or 4 from China, 3 or 4 from Italy, 1 from the UK, and a couple from elsewhere around the globe. And what this panel D and E shows is you're looking at neutralizing titers, essentially, of the antisera. And you can see that every single isolate sampled from different parts of the world was neutralized very effectively. I think that also is extremely encouraging, because there has been some concern that some strains will be neutralized and not others. So far, it's looking very good in that, to that level. Next slide, please. So that's very good news with this inactivated viral vaccine from China. Also, good news has come out recently from the vaccine being developed by the Jenner Institute at Oxford University in the UK. This is a different kind of vaccine. Instead of growing the virus and inactivating it, they put the gene encoding the spike surface protein of COVID into a disabled replication defective chimpanzee adenovirus. So they're using a harmless virus to actually give to people. The virus gets in, it cannot replicate and produce new virus, but once it gets in, all the proteins of that defective genome are expressed, including the spike protein that they inserted into it. And what they've shown is that they have obtained a good immune response from 1 dose. The Jenner Institute have been working on this for several decades in the context of developing vaccines to many pathogens, including malaria. And so this kind of vaccine has been tested in people for 10 different diseases involving thousands of clinical trial participants, and it has been shown to be safe. As of today, no vaccine using this technology has been approved, but I think the Oxford work is looking very promising. And recently it was announced in the media that monkeys, rhesus macaques, that they immunized in Montana in the USA were able to be protected against challenge. Next slide, please. Unfortunately, that monkey data has not been published. So I'm giving you a quote on this slide from the authors, which you can read, but 6 monkeys receiving this vaccine were not stricken with the disease and remained coronavirus-free 28 days after a substantial exposure to the virus. So looking forward to seeing the details of that, but on the basis of this data, Oxford have teamed up with AstraZeneca, a big pharmaceutical company, and they're in phase 2 efficacy studies, efficacy trials right now in humans, and they're planning to scale up production into many tens of millions of doses. There's a very similar approach to this is being used by another company in China, CanSino Biologics. Rather than using a disabled chimpanzee adenovirus as the vector to deliver the spike protein, this group uses a adenovirus 5 serotype expressing the spike. And this is already in phase 2 clinical efficacy studies in Wuhan in China. It was announced just a couple of days ago that the National Research Center in Ottawa have formed an alliance with this company to advance manufacturing. And the intention is for the NRC facility in Ottawa to take part in manufacturing the vaccine for Canadians. Also, Johnson & Johnson, a big pharmaceutical company in the US, they are developing a similar vaccine to CanSino as well as to the Oxford group, using a disabled vector to deliver the spike gene and the protein. Next slide, please. And now I'd like to turn to really quite a new approach to human vaccinology, which has been developed by a company called Moderna in Boston. They've teamed up with Lonza to perform large-scale manufacturing. And also Pfizer, another huge company, has teamed up with a company in Germany, BioNTech, to produce this kind of vaccine. And as you can see, it's quite a different vaccine. It involves RNA encoding the spike protein, which has been formulated into a lipid nanoparticle, so a so-called LNP RNA. This is really quite a new technology. Next slide, please. Around 20 years ago, the concept of using RNA as a vaccine was first discussed because it was found that naked RNA actually was taken up very efficiently by antigen-presenting cells of the immune system. And you didn't need much RNA to actually get presentation of the encoded protein. I think the problems were stability. It was hard to actually stabilize the RNA. And also there were some issues in actual expression of the protein from the RNA. It seems like formulating the RNA now into these lipid nanoparticles is solving a lot of those problems. So one of the things that they found is that this kind of RNA vaccine is a very potent activator of the innate immune response via toll receptors, RIG-I and MDA5. And that led to inhibition of mRNA translation and antigen expression. And so part of the discovery of the lipid particles is to use uridine nucleosides uridine nucleosides have been replaced with naturally occurring base modifications such as pseudouridine and 5-methylcytidine. When those changes are made, the innate immune system is not activated anything like as much, and that leads to increased translation and antigen expression. Now, it's a very new method, as I said. No vaccine has been approved yet for human use, but encouraging phase 1 clinical data has been shown and published for pandemic influenza. The NIH is sponsoring phase 1 trials for the COVID vaccine. And the head of the NIAID, Tony Fauci, was quoted as saying the data from the phase 1 trials was very encouraging. And so they've started phase 2 efficacy trials. And so it's really moving fast. And the US government has donated $500 million to Moderna to manufacture many hundreds of millions of doses. Next slide, thank you. So it's a fascinating approach, and really this could be a whole new way to make any vaccine, not just COVID. So I wanted to spend a little bit of time discussing some of the details. What they do is typically you make a plasmid DNA from bacteria, which contains the spike gene. And the plasmid is linearized with a restriction enzyme, and then it's copied in the test tube into RNA using T7 polymerase to transcribe the RNA. The RNA possesses a 5' and 3' untranslated region, as well as a poly A tail to stabilize the RNA. The RNA then is capped, so it's put in a citrate buffer, and then a cap on the 5' end, S-adenosyl methionine, is added to methylated capped RNA. So this puts a so-called cap1 onto the end of the RNA, and that increases mRNA translation efficiency. Next slide, please. And just for the lipid specialists in the audience, the lipids are four. There are 4 different lipids. Ionizable lipid, no other details were available when I was doing PubMed. A phosphocholine derivative, cholesterol, and then a PEG, polyethylene glycol lipid. So these lipids are combined at pH 4 and citrate buffer, and then the RNA is added, and then a microfluidic mixer is used to create the actual particle. The particles are then dialyzed against PBS, and then the mixture is filtered to get rid of any bacteria through a 0.2 micron filter, and then stored at 4 degrees until use. And of course, the formulations are tested for endotoxin, and the particles are found to be approximately between 80 and 100 nanometer in size. And 90% at least of the RNA is encapsulated. So these particles actually look like a virus to the immune system. And phagocytes and antigen-presenting cells are there to recognize the virus and take up that virus efficiently and present it to T cells and B cells to make an adaptive immune response. So this is part of the reason this approach seems to be working very well, as far as we can tell. Next slide, please. So what happens to these particles? You know, because it's a new method and hasn't really been studied very well in animals or in humans, of course, I wanted to include this slide where they took mice and they injected 6 micrograms of the RNA vaccine into each mouse. So as you can see, the dosage is 300 micrograms per kilogram, which is fairly low. So where does it go to? What you'll see on the right-hand side is that at the injection site, there's the highest concentration of the vaccine, as you would expect. But also, as you would expect, it also ends up in the lymph nodes, in the proximal lymph nodes. There's a heavy concentration, which is good. And also in the distal lymph nodes, there is an elevated concentration, as you can see. Where else does it go? It ends up in the liver and also in the spleen. Next slide, please. And by the way, the half-life of these particles in those organs is around 24 hours. Sorry, next slide, thank you. So, excuse me. I haven't seen any data of the COVID vaccine yet in animals from this group. but I looked at what else they published in animals and in the clinic, and they've done a lot of work previously to COVID on influenza vaccines. And specifically, they've looked at pandemic avian influenza strains H7 and H10. And here on this slide, you see the results where they immunize mice with a H7 mRNA lipid formulation. It encodes hemagglutinin, which is a major determinant of neutralization when antibodies are made to it. And what they've shown is that this vaccine achieved a rapid and sustained protection in mice. So if, for example, you look at panel B here at the top, you can see that all the animals that received a dose of vaccine, whether it was 0.4 micrograms or 2 micrograms or 10 micrograms, survived. 100% of the animals survived. In contrast, the placebos and the controls died at quite a rapid rate. Most of them are dead by day 10. So very effective in mice. The next slide, please. And once again, we're looking at protection against influenza, not against COVID in these slides. They then tested this influenza vaccine in ferrets, and they used mRNA encoding the pandemic avian H10 hemagglutinin. And essentially, as you see at the top, the yellow and the turquoise points are indicating that they got very high levels of HAI. So these are antibodies that inhibit the virus-induced hemagglutination of blood cells, erythrocytes. And this is a correlate of protection for influenza, this assay, HAI. And you can see they got tremendous levels of antibodies in the ferrets using this method, just using 50 micrograms or 100 micrograms of vaccine. Thank you, next slide. Then they tested this influenza vaccine in non-human primates, and they either immunized the primates intradermally or intramuscularly. And they used either 200 micrograms or 400 micrograms of vaccine, which is quite high levels. And they gave them to males and females, either IM or ID, and they immunized twice on day 1 and day 22. And then they looked at the HAI titers, the antibodies that inhibit hemagglutination, and you can see, once again, very high titers were induced with these vaccines in non-human primates, getting titers of up to 10,000 after just 2 immunizations. Next slide, please. Okay, well, this particular influenza mRNA vaccine, they did test in a phase 1 trial. The initial data was published in 2017. Comprehensive data was published 2 years later. I'm showing you the early data, which is still pretty good. So here we're looking at what happened in individuals who received placebo or 100 micrograms of the influenza mRNA vaccine. And you can see in terms of individuals receiving the vaccine that produced HAI titers greater than 40, which is often associated with protection to influenza, you can see 100% seroconverted. 78% of the vaccinees made HAI titers greater than 4 times the baseline. So there was a very nice seroconversion event, and this was very good to see. And then over here on the right, this is, you're looking at the percentage of patients that were positive in a micro-neutralization assay of the virus. And you can see that 87% actually generated neutralizing antibodies. And this, as you can see, is with 100 micrograms of the mRNA vaccine. So if we look down in panel C, you can see that the placebo in turquoise, as you would expect, did not generate any HAI titers, whereas the vaccine here generated such antibodies in nearly all of the individuals, and the median titer was approaching 1 in 100. And over here on the right at the bottom, you're seeing neutralizing antibody titers in the vaccinees. The median titer was around 1 in 50, and the placebos are shown in turquoise. So these titers may not seem that much, but they are generally above the background that is considered protective against influenza. And you know, this is really very promising data, I would say, with the influenza vaccine, and we hope to see similar data with the COVID vaccine. Next slide, please. Yeah, once again, it's a new vaccine, so we have to be very careful to make sure it's safe. They looked thoroughly in this influenza trial, and I don't want to labor the point, but at the bottom bullet point is really the summary. The reactogenicity profile is very similar to what they've seen with more conventional flu vaccines, adjuvanted AS03 adjuvanted vaccine, and the reactogenicity profile was comparable to that seen with meningococcal conjugate vaccines in healthy adults. So nothing is really showing up safety-wise in this small phase 1 trial. Next slide, please. Yeah, so just to conclude the RNA vaccine section, it obviously is looking very promising. It's a tremendous method in terms of rapidity. The vaccines can be made very fast. They can be made at very high dose. And I presume multiple doses can be given to humans. And I think as the NIH is concluding, right now it's looking a very promising vaccine to COVID. Now, what other kind of vaccines are there? There are DNA vaccines. Excuse me. So DNA vaccines emerged maybe 20 years ago, and very good data was obtained in mice and ferrets. Those animals could be protected against influenza with naked DNA. Unfortunately, that never really successfully translated to humans. They just were not antigenic enough in humans. The other sort of issue also with DNA vaccines is that they can integrate into the human genome, albeit at a low level. When that happens, you could activate an oncogene or you could inhibit a key host protein or other interactions. So there are some safety concerns using naked DNA or DNA vaccines in general in a prophylactic mode. Now, the potency has been somewhat solved, I would say, recently by electroporating the DNA into the muscle of the recipient. So there's a needle that injects the DNA, but around that needle are typically 6 electrodes. So as the DNA is injected, the needles also obviously give an electric current, and you're forcing the DNA into the myoblasts and also into APC, antigen-presenting cells at the site of injection. It sounds pretty horrific, but Inovio, the company that's developed this technique, has got it down now where apparently you just feel like a slight itch when the current is applied. DNA vaccines are very good at eliciting CD8 positive cytotoxic lymphocytes, not so good at T helper responses and antibody responses. But the Inovio electroporated DNA vaccine is in clinical trials as we speak with COVID. Now, John Lewis, our own U of A professor, he has worked with Roy Duncan in Dalhousie, and their way of solving the potency problem of DNA is to do something very similar to the Moderna RNA technology, and that is to actually formulate the DNA vaccine into a lipid nanoparticle. It's obviously proven very successful for RNA vaccines. and I think their approach is very promising to enhance the potency of DNA vaccines. Not only that, but they've also included a fusogenic peptide from a virus. I think respiratory syncytial virus, for example, you can include a short peptide into this lipid nanoparticle, which actually enhances fusion of the particle with cells, including antigen-presenting cells. So this is a very exciting approach from John and Roy, and they are in the process of analyzing animal immunization data right now. And John has successfully applied it to cancer therapy, where it's a great way to express various proteins that might be anti-oncogenic, and he has had success in that area. And we're hoping that this works well for the COVID vaccine. And there is a Canadian company formulated around this technology called Entos. Next slide, please. Now, what we're doing in the Li Ka-shing Applied Virology Institute is really what I would refer to as the tried and trusted technology. This is actually making the spike protein in mammalian cell cultures. We use mammalian cells because we get good folding of the protein. It's a very big protein, the spike protein. It's highly glycosylated. And mammalian cells have all of the host factors required to fold the protein correctly into its native state. And this kind of technology has been used very successfully for decades in the context of the hepatitis B vaccine. The surface antigen of hepatitis B is made in yeast and extracted, an adjuvant is added, and it's an extremely effective vaccine for Hep B. Also, it's been applied to papillomavirus, and is now a very good vaccine to protect individuals against papilloma. And more recently, a very good vaccine has been developed to prevent shingles, and it's a highly effective vaccine to do that. And of course, it's been applied many times to bacterial vaccines as well, for example, meningococcal vaccines. So, basically make the key protein, which is the target of the immune system, add an adjuvant, and when you immunize people, you will get neutralizing antibodies that prevent infection by the virus, along with T-cell immunity as well as a result of presentation from the adjuvant. So I think, most likely the correlates of immunity for COVID will be neutralizing antibody titers. And I think this relatively old technology probably will turn out to be the most potent method to elicit the highest titers of neutralizing antibodies. And also, that's important in the context of duration of immunity as well, because the way the immune system works is the stronger the prime, the higher you prime B cells and T cells with your vaccine, the higher memory responses you make, such that when the individual is exposed several years, even a decade or two later, there's a strong memory response still residing in the body that can then quickly amplify to protect from a delayed challenge. So getting the highest titers possible is very important, not just to increase the potency of people that might be seeing the virus soon after immunization, but also in the long term, whenever they're challenged with the virus, obviously by accident. So we think this vaccine will be a good standalone vaccine. It takes longer to develop than the RNA and the DNA and the vector vaccines. So we think another way we can use our vaccine is actually to boost recipients that have already received an RNA or DNA or vectored vaccine. And I think we would anticipate boosting the neutralizing antibody titers with our booster immunization with our vaccine. Next slide, please. So what we're doing is we're in the process of making the spike protein of COVID in mammalian cells. There are some tricks around doing that, And what we're aiming to do is to make a trimer of the spike protein, shown here on the left. This is a cryo-EM structure solved in Texas recently of the trimer. And it's very interesting because this is a native structure that's formed, and there's a receptor binding domain in each molecule of spike. But in the trimer, only one of the receptor binding domains shown in green is actually accessible to bind to the receptor, which is the angiotensin converting enzyme 2. The other receptor binding domains of the other two molecules in the trimer is actually inaccessible, termed closed or down. Over here on the right, you can see where the molecule, the trimer, has been inverted. The ribosome binding domain that's accessible here in green is accessible to the receptor, but the other two are actually closed. And what's thought to happen is when the trimer engages ACE2, there's a sequential opening up of the other receptor binding domains, almost like a bloom flowering. That's the hypothesis. And so we're making this trimer in Chinese hamster ovary cell lines. In order to get a vaccine into humans, you have to use a qualified cell line. So we use a CHO cell line that we obtained from the American Tissue Culture Collection. It has a very good history, documented history of not being exposed to any serum that might have contained bovine spongiform encephalitis, mad cow disease. And it's got a very good record of quality associated with it. So on the next slide, please. What we're doing is we're using the technologies that we've developed over the years in Alberta for our hepatitis C vaccine and applying them to COVID. And we have a very large team. Some of them are from vaccine companies previously. So we have 2 people expert in bioreactor growth of CHO cells. Here you see one of our 12.5 liter reactors. We have 4 or 5 of them and many 5 liters. And we anticipate taking the media from these cells, concentrating them by tangential flow. And Mike Logan and John Law in the lab on Hep C, they figured out a way to produce a very high scale manufacturing level purification method. And what they did is they put an immunoglobulin domain, the constant region of immunoglobulin, they fused it to the surface protein of HCV. And we end up with a precursor being made in our CHO cells. And that enables very large scale purification on a protein A affinity resin. This is the kind of methods that the therapeutic antibodies companies use. And there are many therapeutic antibodies approved for human use. And then what our team has done, we have a very excellent team of bioreactor specialists, purification specialists, QA and QC specialists, and we have a regulatory consultant from Health Canada. What we've done is our team's been able to figure out a way to make a protease under GMP, and we insert a protease cleavage site here, and then when we trickle the protease through this column, out comes the antigen without the FC. We don't want to immunize people with FC domains, and we have to make sure we've got rid of all of it. So these techniques are essential, really, if you want to make not only a vaccine for humans in clinical trials, but to manufacture a vaccine and deliver to millions of people. So we're in the process of doing this for COVID. Next slide, please. We couldn't do that unless we have a first-rate GMP facility, and we have one at the University of Alberta. It's called the ACTM, the Alberta Cell Therapy Manufacturing Unit, and it's a real world-class facility. It's set up to really make reagents for clinical trials. It wasn't really set up to be a production facility. Greg Corbett is the head of this facility. Gail Piat plays an important role in running this facility with her team. And we've had a very good collaboration with them with our Hep C vaccine, and we're planning to use their facilities to make the COVID vaccine. And as I mentioned, we have a very large team in our institute that go into the ACTM and work with Greg's team and Gail's team to make these vaccines. So we're very grateful to have this facility available. Next slide, please. Timelines. Everyone's interested in timelines, of course. We are aiming to be in the clinic later this year. We didn't, we got our CHR grant March 31st. So we've only been going not even 6 weeks yet, but we're going to accelerate it as much as we can, and we hope to be in the clinic in phase 1 by the end of the year. As I mentioned, this technique has been shown to be safe. It's certainly one of the best ways to elicit neutralizing antibody. And although Greg's ACTM facility was designed for clinical products, for clinical trials, we could probably use it for production. We've estimated we probably can manufacture several million doses for Canadian recipients. And we think, hopefully, we're sure Health Canada will be helping us to accelerate the vaccine into the clinic and for human use. And I think there's a good chance we'll be able to roll this vaccine out next year into high-risk groups who are healthcare workers, elderly, and people who are infirmed with cofactors for COVID disease. Next slide, please. I just want to tell you about some other activities ongoing in Canada, elsewhere. VIDO in the University of Saskatchewan, they're very active. They're also working on a subunit vaccine like us with the recombinant spike antigen. They have a variety of adjuvants that they've been working on for many years, and they want to apply, excuse me, those adjuvants to combine with their recombinant spike protein. They've done a great job. They've already developed hamster and ferret models of COVID virus infection. And before you can put a vaccine into a human, you must have shown that it actually works in an animal model. And VIDO have been able to infect hamsters and ferrets with the COVID virus, and they detect viremia, of course, and they also detect some of the disease in the lung that you see in humans. So it's a nice disease model, at least a partial disease model. VIDO recently was the recipient of $23 million from the federal government, and that's good. That enables them to complete their manufacturing facility, their GMP facility, and also to maintain their high containment laboratories, because when you do animal models with COVID, you need BSL-3, which is a very high containment, and it's a very expensive facility to run. Next slide, please. 2 days ago, it was announced that the National Research Centre of Canada in Ottawa is going to team up with CanSino. CanSino, as I mentioned earlier, has a adeno5 vectored vaccine to COVID. And NRC are planning to do some GMP manufacturing later this year in Ottawa in order to test and provide some of this vaccine to Canadians. Next slide, please. And then there's a company in Quebec City, Medicago. One of their specialities is to make vaccines from plants. And what they've shown, as you can see here on the right, is a viral-like particle that they're able to make from the COVID virus in plants. And on the left, you're actually looking at the virus itself. And you can see that the viral-like particle that Medicago makes, you can see the spike proteins here sticking out from the surface of their viral-like particle. And it's very similar to the spike proteins actually protruding from the virion itself. So this is a promising vaccine, and they're scaling up for human trials later this year. Next slide, please. I want to end with this question because I think it gives us some lessons on what we need to do in the future. So the question is, when we experienced the SARS epidemic of 2003, which we now call we now assign that to SARS coronavirus 1, but the epidemic was in 2003. Many people, including the group I was with at that time, developed a vaccine to coronavirus 1, but we developed it quite quickly within a year, but then the epidemic disappeared. And then companies around the world stopped manufacturing it, and they didn't stockpile it. So the question is, if we had have stockpiled that vaccine against the original SARS in 2003, would it have prevented the COVID-19 epidemic that we're experiencing with great tragedy all around the world? And I think the answer is probably on this slide here. This was a paper published from Alexander Wars and colleagues just a few months ago. And what they did is they made the spike protein of the coronavirus associated with SARS in 2003. In the way that we're doing for the coronavirus 2 causing the COVID disease. And then they immunize mice. They immunize mice with this protein, with an adjuvant. And then down here, they took 4 mice and they tested them for their ability to neutralize the 2003 strain of the coronavirus. So they're using what's called a pseudovirus. I don't want to get into the details of that, but it's a neutralization assay using a pseudovirus, but essentially it's a correlate of virion neutralization. And you can see in terms of the control, you get 100% infection. And what you see here is that each of these 4 mice was able to very effectively neutralize the SARS 2003 virus, obviously after vaccination, with this trimeric spike of coronavirus 1. Now, what happens if you try and neutralize coronavirus 2 associated with COVID? Well, here's the control, 100% infectivity in cell culture. We're looking at cell cultures. And now the same mice that were tested here to be totally effective against coronavirus 1, you can see they don't inhibit the infectivity as well, but they do markedly inhibit the infectivity of the COVID virus by 90%. Each of these 4 mice inhibited the infectivity of the COVID virus in cell culture 90%. I think what that means is that if we had have stockpiled the SARS 2003 vaccine, we probably could have prevented most of the disease and the fatalities that the world is experiencing. And so that leads to one important lesson from this epidemic. Next slide, please. This is my slide, my final slide on prospects and issues. For us to manufacture our vaccine under GMP, to do all the things you need to do to satisfy Health Canada, which are good things to do to make sure it's clean and safe and highly tested, to do the preclinical toxicity testing and the product manufacture, and to get it into phase 1 clinical trials, we do need funding, which we've applied to CHR for earlier this week. I think what will happen later this year is that we'll start to see vaccines approved for COVID use, and in high-risk people probably first. And I think it will be the RNA vaccine, I think it will be the adeno-vectored vaccines, and I think the inactivated vaccine also from China. So we'll start to see these vaccines approved towards the end of this year, I believe. Personally, I think they're going to be effective, but probably not as effective as our subunit vaccine that we're working on. So I think one way that they can all be combined, as I mentioned earlier, is that when our vaccine's available, I suspect if we immunized recipients of the other vaccines, we will boost neutralizing antibody titers, increase memory responses, and give a very good vaccine for many years of protection. NRC, as I mentioned, will be producing the adeno vaccine in their facilities later on this year for human use. One thing I think Canada needs to do is to bring in the technology for the RNA vaccines. Thanks to NRC, who are bringing in the adeno technology, which is looking very promising. I think we need to figure out a way to bring in the RNA vaccine technology from Moderna and from other companies that are working on that. Simply because many of the vaccine efforts in Canada are probably going to be more potent, but they take longer. And we need something, and we hope other countries that develop the RNA and the adeno and the inactivated virus vaccines will share those vaccines with Canadians, and they may well do that. But we ought to play safe and also get that technology and start working on it and manufacturing the RNA vaccine. So I think we've also learned, I think, that in terms of dealing with a pandemic like this, Canada really ought to have its own large pandemic vaccine manufacturing facility. Canada can probably make enough vaccine for its citizens, but it will have to be made at different sites all over the country. Our site, or I should say Greg's site in ACTM, the NRC sites, Vito sites, perhaps Medicago, who knows, and other company sites. We'll have to use lots of them. That's not the ideal way to make a vaccine for a pandemic. You really want a large facility that can make enough vaccine for the whole country in one place, because it's easier to get all the vaccines approved by the regulators. So I think this pandemic is kind of teaching us several things. And one is, I do believe we should consider having a large vaccine manufacturing facility in Canada to deal with future epidemics. And unfortunately, we know only too well that from the history of infectious disease, that every few years we have a major virus emerging. In the last 40 years, we've seen HIV, we've seen SARS 2003, we've seen pandemic flu strains, we've seen West Nile virus. So unfortunately, one thing we can be sure of is that future epidemics will arise. We were all expecting it to be a pandemic influenza strain. Turns out to be a pandemic coronavirus, but we're going to have more of these in the future. And I think one thing we've learned is we could use a larger manufacturing facility in Canada. I think the other thing we've learned is that when we make these COVID-19 vaccines and when we make pandemic influenza vaccines, we better stockpile those, because when a new virus emerges, we might be able to at least partially neutralize and protect against the new emerging viruses. Next slide, please. And that's it. I'm very happy to take any questions. Thank you for your attention.
Thank you so much, Mike. Okay, so people have been busy in the Q&A section. And I think one thing that's come up quite a lot in the questions, Mike, has been for a little bit of understanding and clarity about what the phases are for testing and developing of vaccines. So when you refer to the different phases in testing in animals, what the practicalities of that are?
Yes, that's a great question. Traditionally, clinical development to get a vaccine approved for human use goes through at least 3 phases, 1, 2, and 3. And it can often take 10 years. It's not unusual. In fact, it's almost commonplace for the whole process to take 10 years before you're allowed to deliver it to humans. With COVID, because it's such a dreadful disease affecting so many people, the regulators will speed it up. I'm hoping that when we get our initial clinical data next year, early next year, if we can show that we're getting good neutralizing antibody titers and it's safe, which I think it will be, because we've had a lot of experience with these kinds of vaccines, I'm hoping the regulators will say, okay, antibodies are the key correlates of immunity. you've demonstrated safety, will allow you to cautiously roll it out now into high-risk individuals in Canada, healthcare workers, elderly people, people with disease. And so I'm hoping that that's the way it will happen, which will be very, very fast. Normally it takes at least 6 and often 10 years to get a vaccine approved for human use.
Mhm. Yes, thanks, Mike. So there's a follow-on question to that from Dr. Vora. And she asks, exactly that given it does take quite a long time, and, what do we know about safety profiles of some of these different approaches? And she asks at the end, wouldn't adapting known vaccine technology approaches be safer than something that's novel, both in technique as well as content?
Yes, I mean, tried and trusted technologies would be safer. We know they're safe, adjuvanted proteins. Inactivated viral vaccines, we have a lot of those approved for human use. The new ones, the RNA and the DNA and the adeno, we don't have such a safety record yet built up. But obviously, we desperately need a vaccine out there as quickly as possible. So I think the regulators will be willing to take more risks at approving a vaccine than they would otherwise, because the risk to benefit ratio is very different from most other vaccines. I think with the RNA vaccine, obviously hardly any history so far in humans. We need to see what happens after immunizing a few thousand individuals. The DNA vaccine, there are some issues around that in terms of integration into the genome and maybe activating an oncogene and so forth. And I think that's why the technology John Lewis and Roy Duncan are developing might help a lot, because it might reduce the required dose of DNA to reduce the risk of integration. And then the adeno, I think the adeno is going to be safe. It's a completely disabled vector. It cannot produce new virus, but it can express the spike protein very well. So I think, in terms of safety, I would put the adeno vector, adjuvanted proteins would be the top of the safety list, followed by inactivated viral vaccines, followed by adeno vectored vaccines, followed by RNA vaccines, and then DNA vaccines, which isn't to say that DNA vaccines won't get approved, but there'll be more issues. The first priority, obviously, is to protect the people at high risk, healthcare workers. We've got to protect our heroes in the front line. We've got to protect the elderly. As we all know, they're suffering very, very badly. So I think you can take a bit more risk with a vaccine for those groups. But if you're talking about vaccinating 33 million Canadians, including children, that's where you have to be very careful. That's where you need to build up the safety database in 20, 30,000 people before you start vaccinating millions.
So given that preamble, Michael, when you think about the lipid nanoparticle work and the RNA work, is there any other advantages? Is it faster to produce? Is there some other, is it less expensive? Are there other reasons why some of these are being pushed at the moment as well, that might be a reason to think about them?
Yes, thank you, Denise. Yes, the RNA approach and the DNA approach, very rapid, relatively easy to make the vaccine in high levels, high amounts. That is a big advantage. And as I mentioned, the RNA technology has just evolved very recently, and it's already giving some pretty good data in humans with the influenza vaccine. So speed and scale up are two big advantages of the RNA vaccine approach. The negative is we don't have any experience of using it in humans, or hardly any experience, just apart from 100 or 200 individuals that received such a vaccine.
So Mike, we have some questions about, you had mentioned about the SARS-1 vaccine, and yes. Barbara was asking the question that if this vaccine was developed or exists, would that be something to think about doing quickly, to try to provide some partial protection, or is there any merit to that?
Yes. Well, like I mentioned, it's too bad we didn't stockpile the SARS 2003 vaccine. But I think now we'll be better off making the COVID vaccine. There does seem to be neutralization, cross-neutralization of the two viruses. But if we're going to start manufacturing anything, it would make sense to do the COVID, because then instead of getting 90% neutralization, we should get 100%. So I think that's what we should do, focus on COVID.
And Nandini has a question here about whether there's any evidence that BCG vaccination is protective at all to COVID-19.
Yeah, that's a great question. I think the answer is it wouldn't be a prophylactic vaccine, but several people around the world are suggesting that BCG, polio vaccines, anything involving a attenuated replicating vector, bacterium or virus, might be therapeutic. And that's because if you gave it to an acutely infected patient, the actual act of introducing a replicating entity into that person will stimulate cytokine release, will stimulate the innate immune response as well as the adaptive. It will kick the adaptive immune response up a notch. And there is some data suggesting that could work. So, you know, until we find something that really works as a therapeutic, I do think it's worth looking at BCGs and polio viruses. Anything, you know, many vaccines that are used in humans today are attenuated, but they replicate. And when they do that, they activate the immune system, not just to that particular pathogen, but to any other pathogen that's in the body. So I do think there's definitely some merit in administering those kinds of vaccines once people are infected. And there might be an amelioration of the COVID disease when you do that.
Great. And there was a little bit of confusion in the questions around the, as you mentioned, that increased immune response that sometimes can happen when people are vaccinated, and that's potentially a risk. Could you go back over that a little bit and explain to our listeners a little bit about that phenomenon and why we care about that?
Yes, no, it's a great question. So there's a coronavirus that sickens cats. I think Dr. Lorne Tyrell discussed it last week with his colleagues in terms of having a drug that actually can cure those cats. And now Lorne and his colleagues have shown that it can actually inhibit COVID in cell culture. So it's really great news in terms of a new therapeutic. But when people try to make a vaccine to that cat coronavirus, rather than preventing disease, they actually enhance disease. So a very bad outcome, obviously. Now, there are two kinds of enhancement of disease. The first kind is when the antibodies that you make with the vaccine are not very good neutralizing antibodies. They're not very good at neutralizing the infectivity of the virus. They're at very low levels. And unfortunately, there are other antibodies that the vaccine makes that enhances infection. So when those antibodies bind to the virus, they then enable the virus to get into macrophages and monocytes. And once they get in through FC receptors, the virus can replicate at high levels and actually enhance disease. So that's called antibody-dependent enhancement. And the other kind of side effect that people have seen with a vaccine to the cat virus, and also I would add to the coronavirus 2003, if you use a vaccine and you tilt the immune response in the Th2 avenue, in other words, you don't activate T cells to make inflammatory cytokines. You tilt them towards what we call a Th2 response. There has been a phenomena in which those vaccinees develop eosinophilia in the lung, that you get the eosinophils migrating en masse into the lung, and you can get a kind of immune toxicity as a result of the vaccine. So that's why I'm really happy to have seen the monkey data from Sinovac, which not only protected, but also they saw no signs of the Th2 type of toxicity I'm talking about. And reportedly, the Oxford group also are seeing prevention without any immune toxicity. Now, doesn't mean to say it can't happen in the future, and it might depend on the vaccine that's used. But certainly, I know the regulators have basically concluded and advised, if not insisted, quite rightly, that if you're going to use an adjuvant, you better use a strong Th1 stimulator, not a Th2, to avoid anything like the cat vaccine.
And can you just explain to everyone exactly what an adjuvant is?
Yes, an adjuvant is there to really increase the immune response to your vaccine. There's different kinds. Some act as a depot for the antigen to enable antigen-presenting cells time to migrate to the site of injection, take up the vaccine, and present to the immune system, the B and the T cells. That's the old-fashioned type of adjuvant, alum, for example. That's essentially what alum does. It's a depot enabling better antigen presentation. The more modern adjuvants have actually ingredients that directly activate the immune response through various receptors, toll receptors, and also have various ingredients to actually deposit the vaccine onto the MHC class 1 presentation pathway, so that once again you get better presentation, you get a better T cell response and a better B cell response. So it's a combination of depositing, keeping the vaccine around in the body for longer, combined with specific ways to activate the immune system in the local environment of the vaccine antigen.
Thank you. So I think we're going to give Margaret Russell the last question, and her comment was institutional memories are very short. In 1999, an advisory committee recommended that the Canadian government support a sole Canadian manufacturer of the influenza vaccine, and that didn't happen. And so I think this gets into the point you were making at the end, Mike, and I just, in these low probability, high impact events like we're living now, I wonder if you could comment on that, and perhaps we can wind up on that theme.
Well, you know, the old adage that every cloud has a silver lining. It's hard to find one with this terrible epidemic, and it's really terrible to think of all the people suffering around the world. But if there's one good thing that's come out of it, I think, is that it's told us we better get a large-scale GMP manufacturing facility in Canada, because for sure there will be other epidemics like this, and COVID really has been our educator. I think we need to get one. Secondly, we need to stockpile these vaccines for COVID-19. Thirdly, we need to stockpile some influenza vaccines against pandemic strains, because when one of them comes out, we're going to have a repeat of the COVID situation. And we better stockpile some vaccines that we think have some scientific basis for thinking there'll be at least an amelioration of those epidemics, if not a complete prevention. Yeah, so, you know, I think this terrible scourge has to teach us, and I'm sure it will, that we'll be better prepared next time. But we need to move on it. Once we've got this vaccine out there against COVID, we then need to really work hard to get better infrastructure, I would say. And I'm not, you know, it's not the blame of anyone, you know, until you read about this and see it in movies, right? You can't believe it's actually happening, and it has, and we need to learn from it.
Well, thank you so much, Mike, and really appreciate all of our speakers for the webinar series for coming and giving of their time and helping us all understand better some of the science that's happening and work that's happening here at U of A and at other sites in Canada to address COVID-19. And so would really welcome feedback from anyone if you're able to do so. There's a QR code on the screen right now. And if you have time to scan it and provide us some feedback, and also if you have any other topics that are on your mind that you think would be helpful for us to think about putting on additional webinars, we'd be delighted to hear from you. So thanks once again, Mike, and thanks Dr. Terrell in the background for answering questions. And thank you to everyone who came out tonight to listen. Have a really good evening.